Hello everyone! This section is for bacterial cell structure. After learning this section, you should describe the common bacterial structures and their functions. Structures often observed in bacterial cells are summarized and illustrated in this table. This table shows common bacterial structures and their functions. You can see plasma membrane, gas vacuole, ribosomes, inclusions, nucleode, cell wall, and so on. Note that no single bacteria possesses all of these structures at all times. Some are found only in certain cells, in certain conditions, or in certain phases of the life cycle. This picture shows the structure of a bacterial cell. Bacterial cells are often surrounded by several layers, which are collectively called the cell envelope. The innermost layer of the cell envelope is the plasma membrane. You can see it on the picture, which surrounds the cytoplasm. Cell membrane acts as selectively permeable barrier, mechanical boundary of cell. It is responsible for nutrient and waste transport, location of many metabolic processes such as respiration, photosynthesis, and detection of environmental cues for chemotaxis. Then you will see the cell wall. Most bacteria have a chemical complex cell wall, which covers the plasma membrane. It is responsible for protection from osmotic stress, helps maintain cell shape. In typical gram-negative bacteria, there is periplasmic space between the cell wall and plasma membrane, which contains hydrolytic enzymes and binding proteins for nutrient processing and uptake. In typical gram-positive bacteria, may be smaller or absent. Many bacteria are surrounded by a capsule or slim layer external to the cell wall. They help bacteria resist to phagocytosis and adhere to surfaces, because most bacteria do not contain internal membrane-bound organelles. Their interior appears morphologically simple. The genetic material is localized in discrete region called the nucleode, and usually is not separated from the surrounding cytoplasm by membranes. And you can see ribosomes. Ribosomes and larger masses called inclusions are scattered about the cytoplasm. Ribosomes are responsible for protein synthesis. Inclusions are employed for storage of carbon, phosphate, and other substances. Many bacteria use flagella for locomotion. Fibria and pili help for attachment to surfaces, bacterial conjugation and transformation, trichine and gliadin motility. In the remaining section of this chapter, we will describe the major structures observed in bacterial cells in more detail. We begin with the cell envelope. We then proceed inward to consider structures located within the cytoplasm. Next, the discussion moves outward to a variety of appendages that are involved in attachment to surfaces, motility, or both. Finally, we consider the bacterial endospore, which are survivals under harsh environmental conditions. Okay, that's all for this section. Here is a word list for you.